Thank you very much. So let me start then by thanking Kyoto University and Inamori Foundation for the invitation to come here and speak. It's a great privilege to be here, and I always very, very much enjoy coming to Japan and especially Kyoto. So what I wanted to begin with then is to remind you about what you all already know, namely that our genetic information is stored in the form of DNA, this famous double helical molecule. And the information there is stored in the form of the sequence of these 4 bases or nucleotides, abbreviated A, T, C, and G. And it's stored twice, and that's very important because when a cell divides and a new individual will appear, if this is in the germline, the 2 strands come apart and new strands are synthesized with the old strands as templates. This is a very exact process, but sometimes errors are made or mutations, so wrong nucleotide or base is built in, a mutation happens, and we see the traces of those mutations when we compare DNA sequences in the population. So if we compare DNA sequences from 2 people in this room, we will find a difference approximately every 1,000 bases or so there is a difference. And these differences accumulate as a function of time. So if we add in the chimpanzee DNA sequence, we will find more differences, approximately 1 difference every 100 base pairs or so. And if we want to reconstruct the history of such a piece of DNA, we generally do it in the form of a tree like this, where in this case, the 2 human DNA sequences go back to a common ancestor quite recently in time, and much further back is a common ancestor shared also with the chimpanzee. And as you also know, the human genome is about 3 billion nucleotides or bases long. So there are about 3 million differences between 2 genomes. So there is a lot of information here with which we can try to reconstruct human history. And if you do that on a genome-wide scale and a worldwide scale, what you will find is that you have most variation in Africans today. And if you take the entire world outside Africa, you have less than genetic variation. And not only that, those DNA sequences you find outside Africa generally have close relatives inside Africa. But there is a part of the variation then in Africa that have no close relatives outside Africa. And the interpretation of this is that modern humans, our direct ancestors, evolved inside Africa, accumulated genetic variation there, and a part of that genetic variation, so to say, went out and colonized the rest of the world. And with some genetic tricks that I will not explain, you can also estimate approximately when this exodus out of Africa happened, and it's in the order of 100,000 years ago or even less. So this is the genetic evidence for this recent African origin model for modern humans. But there is a sort of complication with that model, if you like, and that is that there were other forms of humans around since a long time, also outside Africa. Most famously then Neandertals in Western Eurasia, and other forms of now extinct humans in Eastern Eurasia. And the big question is, that have always interested me, is how were these extinct forms of humans related to people who live today? And what happened when one met them? But in order to ask that question, you will then, of course, have to go back and retrieve DNA from such old bones. And that is then the interest of my lab since over 30 years now. And it seemed then in the early 80s when I started this work that the obvious place to start would be ancient Egyptian mummies. They go back 2, 3, 4,000 years, and they look very well preserved. But it turned out that most Egyptian mummies actually contain no DNA you can retrieve. But there are some exceptions to that. This is the first exception, a 2,300-year-old mummy of a child, where in the skin, in the upper layers of the skin, you can see things that look like cell nuclei where DNA would be preserved. And you can also stain DNA in here. And at that time, in 1984, Alan Wilson, a very famous evolutionary geneticist, also showed that they could retrieve DNA from the skin of a quagga, an extinct form of a zebra that was around 100 years old. So I also then retrieved DNA from this Egyptian mummy and found some human DNA in it. But over the years that followed then, it became quite clear that that was very surely contamination. It was probably DNA from me or someone in the lab. So those DNA sequences were clearly due to contamination, but this sort of staining of the DNA in the cell nuclei, after all, showed that there was some DNA there. So what became clear then over the next few years is that there are many technical complications with working with ancient DNA. The first complication is that there's very little there. So compared to present-day modern tissues, in the ancient tissue, you'll have 10,000 to million-fold less DNA. And it's chemically modified, and it exists in a large excess of bacterial DNA from organisms that colonize the tissues after the death of the individual. So this then means that tiny amounts of contamination that will not at all show up if you study present-day DNA may even overwhelm the experiments when you study these old tissues. So there was a number of years where we just focused on avoiding contamination with different technical tricks and with dressing up in funny clothes and working in clean room conditions, etc. And we then focused on the mitochondrial genome, simply for the fact that there are many copies of this tiny part of the genome here. So there's a bigger chance that some fragments of it will survive. Although most genetic information, of course, is in the cell nucleus. We started out by zoological collections that are just 100, 150 years old. Went back to recently extinct animals, such as the marsupial wolf that became extinct around 100 years ago, and to animals that during the last ice age disappeared, such as the ground sloth in America or mammoths. But our big interests have always been Neanderthals. So here is a reconstructed Neanderthal skeleton compared to a present-day human skeleton. The Neanderthals appear in the fossil record somewhere 300,000 or 400,000 years ago and exist until around 40,000 years ago when they become extinct. And there are two different ideas about the relationship of Neanderthals in Europe then to modern humans that come out of Africa. One is total replacement, saying these modern humans come from Africa, replace the Neanderthals with no mixture. Another one is that modern humans come out of Africa, but mix with Neanderthals so that there is a contribution from Neanderthals to present-day Europeans and from other groups in Asia to present-day Asians. So the consequences of this is then in the one extreme, total replacement, 0% contribution from Neanderthals to present-day people. And you can imagine any number of fraction of contribution up to even total continuity. So we got the first chance to test this in the early 90s when we got access to the Neanderthal type specimen. So this is a fossil that was found in 1856 in Neanderthal and gave its name to this group of hominins. Had a sample from the upper arm here, looked for the mitochondrial genome, a particular variable part of the mitochondrial genome, retrieved a piece of DNA from shorter fragments that were retrieved many, many times and believed the changes that were consistently there, and reconstructed such a phylogenetic tree. And you will see that the mitochondrial genomes of present-day humans then go back to a common ancestor 100 or 200,000 years ago, and the mitochondrial genome of the Neanderthal goes back much further, 500,000 years ago or a little more. Since then, we and others have retrieved many other Neanderthal mitochondrial sequences. They all fall together outside the variation of modern humans. So it's very clear there are no humans today that run around with mitochondrial genomes from Neanderthals. So in this set of things, it's then total replacement, 0% contribution. But it's of course clear that the mitochondrial genome is a very tiny part of our genome. The big picture will of course be in the nuclear genome, where we inherit DNA both from our mothers and fathers. But I think until 15 years ago or so, I'm even on the published record of saying that we will never be able to retrieve the nuclear genome of Neanderthals. It will be too little there, it's too degraded, and so on. But what changed all that was the advent of new techniques, high throughput DNA sequencing techniques that allow you to sequence millions of DNA molecules quite fast and quite inexpensively. So instead of trying to retrieve the mitochondrial genome, you can now simply extract all the DNA from a fossil, sequence all the DNA that's in there, make your own little database, and start compare it to the human genome, to bacterial genomes, and so on. And the first place where this worked for Neanderthals was this beautiful cave in southern Europe, in Croatia, Vindija Cave. From this bone here, that's about 40,000 years old, this piece of the bone. And we were then very lucky to receive funding over 5 years to try to reconstruct a Neanderthal genome. So we worked very hard on improving the methods with which we extract DNA from bones and convert it into form that you can feed into the sequencing machines. So we got much better on that. The machines got much more efficient. And what we then find is, indeed, as we already knew, the length of the DNA fragments is really very short here, 40, 50, 60 base pairs. Whereas if you extract DNA from a blood sample of me, you will easily find pieces of 10,000 or 20,000 base pairs. The vast majority of the DNA also is not at all from the Neanderthal, but from bacteria that lived in the bone. Our best Neanderthal bones have something like 4% Neanderthal DNA. We also found that there were particular chemical modifications towards the ends of these molecules, where we find that Cs appeared as Ts in the DNA sequences, particularly towards the two ends of the molecules. And that turned out to be due to a chemical modification of the nucleotide C that loses this chemical group, so it becomes an unnatural base in DNA, U, that is read as T. So we have apparent C to T mutations in the end of the molecules. So we then focused on 3 Neanderthal bones, sequenced a bit over 1 billion DNA fragments from them, and mapped them to the human genome, taking these special types of mutations we see into account at the ends of the molecules. So in 2010 then, we had a little over half of the Neanderthal genome, a first draft version of it. So we could begin to ask questions. And one of the first questions we were interested in was, of course, what about the relationship to present-day humans, to modern humans? What happened when modern humans came out of Africa and met Neanderthals? Did one mix or not? So an expectation, if that would have happened, would be that Europeans today should share more genetic variants with Neanderthals than Africans today, because there have never been Neanderthals in Africa, so there's no reason to assume that they should carry any contribution from Neanderthals. So we then had the Neanderthal genome here, and we went and sequenced the genomes of 5 present-day people from different parts of the world and did a very simple test. We simply compare 2 present-day genomes. To begin with, 2 Africans here, find all places in the genome where they differ from each other. Then we take the Neanderthal genome and say, how often does that match one African or another African? And this should be 50-50, right? Since Neanderthals have never been in Africa, there is no reason to assume that the Neanderthals would have contributed more to one African than the other African, and statistically speaking, that's the situation. Equally many matches to this African as that African. If we then compare a European and an African, to my surprise at the time, actually, I must say, we found statistically significantly more matching to the European person than the African person, suggesting that the contribution from Neanderthals had existed here. Even more surprising was when we take a person from China and from Africa, again we see more matching to the Chinese person, although most people would think Neanderthals have never been in China. And even more surprising, in Papua New Guinea, we again see the same thing, although everyone will agree there have never been Neanderthals in Papua New Guinea. So the model we suggested for this, that has since been borne out by other work, both by us and others, is to suggest that when modern humans come out of Africa, they presumably passed by the Middle East. We know there were Neanderthals in the Middle East. So if these early modern humans mixed with Neanderthals and then became the ancestors of everybody outside Africa, they can sort of say have carried with them this Neanderthal contribution out to the rest of the world, to the extent that between 1% and 2% of the DNA of everybody outside Africa today come from Neanderthals. It now turns out it's a more complicated story than this. There are more events of admixture with Neanderthals, and I'll mention that briefly in a few minutes. So what we have since done in 2010 is to generate a much, much better Neanderthal genome. And that is due to collaboration with academician Anatoly Derevyanko in Russia, who excavate at this site, the cave site, the Denisova cave in southern Siberia, this beautiful place, where in 2010 they found a toe bone that turned out to come from a Neanderthal. And we had by that time developed a much more sensitive way to make DNA libraries from old remains that relies on separating the two DNA strands and then make a library from these single strands. So each double-stranded DNA molecule has two chances to make it into the library, one from each strand, so to say. And this, and some other tricks, allowed us then to go from this Neanderthal genome where we had quite few fragments and many holes, just a little over half the genome covered, to a very good genome, where we sequenced every position over 50 times over, so that every position to which we can map such short fragments are now covered to a quality as high or even higher than present-day human genomes. So you can now look in different present-day people here for one chromosome, for example, and see which parts does a certain individual carry from Neanderthals. As you see, we carry different parts from Neanderthals generally, but the sum of them in any single one person is somewhere between 1% and 2% if your roots are outside Africa. You can also ask how much of the Neanderthal genome exists today. If you add up these fragments, you can sort of puzzle together longer pieces of the Neanderthal genome in people today. And it's a bit unclear how much that will be, but it will add up to something maybe approaching 40% of the Neanderthal genome or so. Anatoly Derevyanko also found another little fragment of a bone in the Denisova cave. It's a part of the last phalanx of a pinky of a child. And when we applied our methods to this piece of bone, we could again retrieve a high-quality genome where each position is sequenced over 30 times over. And to our big surprise, we found that that individual was not closely related to Neanderthals. It had a common ancestor shared with Neanderthals, but far, far back. So we realized we had found a new type of extinct humans, and after thinking a lot about it, we decided to call them Denisovans. So just like Neanderthals are called Neanderthals after Neanderthal, where they were first found, Denisovans are then called Denisovans after Denisova cave, where they were first found. And they are indeed quite distantly related to Neanderthals. So if this is the population, deepest population split between two human groups today, they are in Africa, depending on now the mutation rate, but if we take a mutation rate, this would be in the order of 100,000 years back or so. If we compare Neanderthals and Denisovans, they go back in the order of 4 times further back, their population split, than the deepest split between any two humans today. And just for comparison then, the split between Neanderthals and Denisovans and present-day people is something like 6 or even 7 times further back. So you can then ask for Denisovans, have they mixed with present-day people or their ancestors? And indeed, they have. Surprisingly, although the bones were found here in Siberia, we find the biggest evidence of admixture in the Pacific. So in Papua New Guinea, Aboriginal Australians, and so on. And quite a big contribution in the order of 5% of the genomes or so of people there derive from Denisovans. So if I should summarize what we think we know today then about the origin of Neanderthals and Denisovans, we think that they have an origin probably in Africa, more than 500,000 years ago. They then come out of Africa, and they evolve in Western Eurasia to what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. There are clearly other forms of extinct humans here too, Homo floresiensis and so on, and other forms probably in China. We don't know where the border between Neanderthals and Denisovans were, but we do know that in the Altai Mountains here, at some times there were Neanderthals, at some times there were Denisovans. Then modern humans appear in Africa, come out of Africa, mix with Neanderthals the first time, probably in the Middle East, continue to spread over Eurasia. And there's good evidence from us and from other groups that there are additional admixture with Neanderthals in the ancestors of present-day Asians. People in Asia have slightly more Neanderthal ancestry than people in Europe, about 20% more, so instead of 1%, 1.2%. And we've recently shown also that there was admixture around 40,000 years ago with modern humans in Southern Europe or Southeastern Europe. And there is then a contribution from Denisovans to modern humans in Southeast Asia. And then with time, these other forms of humans disappear, but they live on, if you like, a little bit in the fact that 1 to 2% of the genomes of people outside Africa come from Neanderthals, and another 5% or so from Denisovans in the Pacific. In the future, I would not be surprised if one found contributions from other extinct forms, for example in China, or also in Africa when modern humans spread across Africa. So we've clearly rejected this total replacement model. There is a contribution from extinct forms of humans, but it's generally a small one, less than 10% or so. So the big picture is one of replacement, and there is a little bit of leakage, so to say, from these earlier forms. So we like to sort of suggest a model that we like to call leaky replacement for the origin of modern humans. Now, for the last part of my talk, I wanted to discuss, are there any functional consequences of this contribution from Neanderthals and Denisovans to present-day humans? So you can now go over the genomes of people, hundreds of people in Europe or in Asia, and found places where many people in Asia, say, carry contribution from Neanderthals. Sometimes this is shared with Europeans, sometimes it's unique to Asians or Europeans. And you can ask, statistically speaking, what genes are particularly located in such parts of the genome. And the only type of genes you find there statistically significantly are keratins, so structural proteins in skin and hair. So we'll probably learn in the future that some aspect of the morphology or function of skin and hair in Asia and Europe come from Neanderthals. More importantly, a group in Stanford have shown that immune response genes, the genes important for the defense against infectious diseases, come from Neanderthals and have sometimes reached high frequencies in different parts in Eurasia. A group in China has shown that Europeans, genes involved in lipid catabolism, so degradation of lipids, come from Neanderthals, disproportionately much in Europe, much less so in Asia. So some aspect of how we deal with lipids come from Neanderthals. And quite interestingly, last year one found a risk variant of a gene, a lipid transporter, and this risk variant confers risk for type 2 diabetes. So the type of diabetes you get at old age. And this risk variant is particularly common in Asia and in Native Americans. And it turns out to come over to present-day people from Neanderthals. So it may be surprising that a variant that causes disease today comes from Neanderthals and rises to high frequency in humans. But I think it's a reasonable speculation that variants that give you diabetes today, when you have ample nutrition during your whole life, actually may be advantageous to you in periods of starvation. So it may be that what we have there is some Neanderthal adaptation to starvation that was advantageous to modern humans in the past. It's also known since quite some time that populations in Tibet are adapted to living at high altitudes where there is little oxygen in the air. It was also known that a major adaptation to that was a variant of a gene called EPAS1 that has to do with how many red blood cells you have in your blood. And this variant is very common in Tibetans, 80% frequency, very rare elsewhere, rarely, for example, occurs in Japan and China, but it's very, very low frequencies. And last year, then, Rasmus Nielsen's group in Berkeley showed that this variant comes over to Tibetans, to the ancestors of Tibetans from Denisovans. So it's fascinating to me that we would probably not have such big populations of the Tibetan plateau as we have if we hadn't had this genetic contribution from Tibetans in the past that allowed this adaptation. So there is sort of a picture emerging, and there are more studies like that on the way, that when modern humans spread out of Africa, they come as these sort of newcomers from Africa to Eurasia, and they meet these groups that have lived there for hundreds of thousands of years and have adapted to those environments. They mix a little with these groups, so they fish out genetic variants that are advantageous in those environments, and they rise to high frequency. So it's a sort of phenomenon that we call adaptive introgression that's quite known from some animal species, but have not yet been so sort of thought of in terms of modern humans' origins. So you can also now, of course, start asking what was not contributed to modern humans from Neanderthals, because we can now go across the whole genome, in Europe in red and Asia in green, and see where there are contributions and ask for regions where we would statistically expect a contribution from Neanderthals, but we don't see one, or we see very, very little. So we see sort of holes, if you like, in this. And these regions are very interesting, I think, because they suggest that here is something lying that we sort of positively select, and we select against a Neanderthal contribution there. Here may be something that really defines us as modern humans. So things, genetic changes that are unique to modern humans that happen after we separate from Neanderthals and that we sort of prefer from a selective sense, if you like. Why would that be interesting? Well, I do think that there are some ways in which modern humans are special relative to Neanderthals, Denisovans, and other early forms of humans. One is clearly technology, I think. With modern humans, technology starts changing very rapidly, become regionalized in the world, and of course over 100,000 years or so, our technology has changed in a very dramatic way, allowing us to colonize most of the world and live in very different climatic conditions, etc. Art, at least art that depict things that we immediately recognize as art, comes only with modern humans. And modern humans are, of course, the first hominin that really spread across open water, come to Australia, come to the New World, and colonize every little island in the Pacific Ocean, for example. So there seems to be quite a different behavior here. So a dream is, of course, to be able to go after the biological background for that. And we have the tools for that now, because we can make a catalog of all new features fixed in the human genome since we separated from Neanderthals. So if we are very strict and now look for things that changed here, are present in, as far as we can tell, 100% of all humans today, but where Neanderthals, Denisovans look like the apes. We can make this list, and surprisingly, that list is not very long. It's a bit over 30,000 single nucleotide changes. So you can actually look through them in the computer in an afternoon. The problem is, of course, that we are very bad at predicting any functional consequences of any single position that have changed in the genome. Just to give you a feeling for this, though, there are just 96 amino acid changes in proteins caused by these changes. And they occur in only 87 genes that are encoded in our genome. And this is then extremely interesting, I think, for the next 10 years or so to study these changes. And of course, one will tend to be most interested in genes expressed in the brain, since we may think that in the cognitive area are things that particularly have changed in modern humans relative to other forms of hominins. So one may ask, how should one study in the future these things? And it is a sort of problem, I think, because we're now studying human-specific things. We don't really have animal models. I think practically there are two ways one can imagine to do this. You could introduce these human and Neanderthal-specific changes in stem cells, particularly induced pluripotent stem cells that were discovered at this university, of course, by Shinya Yamanaka, as you are well aware. What you can also imagine to do in the future, I think, to introduce these changes into animal models, such as laboratory mice. And I want to end by illustrating this to you with a gene that we have studied a lot over the past 10 years. It's a gene called FOXP2 on chromosome 7. When you have a mutation in this gene in a famous family in Britain, you have a language problem. So this gene seems to have something to do with speech and language. It has two human-specific changes close to each other that affect the protein that change amino acids. That's unique to humans relative to the apes. So we were, of course, very interested when we started getting information from the Neanderthal genome and the Denisova genome about the state at these positions, these two positions here. If we look at the Neanderthal and the Denisovan, it turned out that they are like modern humans here. So these two changes clearly happened earlier on the human lineage before we separated from Neanderthals. But you can use them then to illustrate how we study such human-specific changes. So they are close to each other in the protein here, so you can then introduce them into a mouse, into the mouse gene. And this protein is very conserved, so they then construct a mouse that essentially produces from its own FOXP2 gene the human protein. And now you then have a model that you can begin to study. And you find, for example, that neurons in certain parts of the brain in these humanized mice with the human FOXP2 genes make longer dendrites, so longer connections to other neurons than in the wild-type mice. And these changes are then particularly prevalent in parts of the brains that we call the corticobasal ganglion circuits, which are connections between basal ganglia and the cortex and the brainstem that has to do particularly with motor learning in humans. So you then have a model where you can start studying this, and this is work by a previous graduate student in our group, Christiane Schreiweis, that worked with Anne Graybill at MIT with these mice and studied motor learning. So they use a paradigm where the mouse has to go in a maze and had to learn to go to a certain signal here to get a reward. And if you teach a mouse always to go towards a signal, and you always have the signal towards the left, it comes a time when you can take away the signal. The mouse has simply internalized that it should always go left to get chocolate, for example. And that switch from when you can turn off the signal, when you automate a motor movement, is done very much quicker in these humanized mice than their wild-type litter mates born by the same mother. So these mice learn in 7 or 8 days, what their litter mates that don't have those mutations learn in 11 to 12 days. So these mice are actually faster in learning certain motor tasks. And this goes together with the shift of activity in the brain from a certain part of the basal ganglia to another certain part that we can also see happening in the mice. So I'm not a neurobiologist, but my neurobiologist colleagues explains this type of learning for me as if you think about when you learn to bike as a kid. When you begin with it, you're very bad. You think about what you do, and you're very bad at bicycling. And then comes a point when you stop thinking, when it has become automated. And that's the point when you get very good at bicycling. And this is this shift from declarative to procedural learning. So the current hypothesis then is that these 2 amino acid substitutions affect something in corticobasal ganglion circuits that allow faster automation of muscle movement. And it's tempting to speculate that the most sophisticated muscle coordination we do in life is actually articulation when we speak. We need millisecond control of our vocal cords, our tongue, our lips to produce articulate speech, and it's something very unique to present-day humans. So a hypothesis analysis has something to do particularly with production of speech. And I think that this is quite hopeful actually, that you can sort of maybe in the future introduce several of these changes that are human-specific into mice, maybe even humanize aspects of the biology of the mouse to study the effects of these changes. But I think it will also be very, very important to do this in stem cells. So to wrap up then, I think over the last 30 years we have come a long way. In 1984, the first papers on ancient DNA were published, and for a number of years there were just 1, 2, 3 papers published. And since a number of years there have been sort of a steady increase in this field. The question I often get of is, how far back do we think we can go? Can we look at much older hominins, for example? So far, almost everything or everything we discussed was in the last 100,000 years or so. The world record in old genomes is actually held by Eske Willerslev, that will give them the next talk here. It's a horse genome that goes back about 700,000 years. But that's then in the permafrost, it's frozen. So if we look in remains that are not frozen, we work since a couple of years now at a site that's 430,000 years old in Spain, Atapuerca, a deep cave site where one finds many remains of probably ancestors of Neandertals or Denisovans, where we've been able to retrieve a mitochondrial genome and now a bit of nuclear genomes. So I think you can, with these remains, at least go back something like 400,000 years outside the permafrost. So I think it's quite hopeful actually that in the future we might be able to go back further, perhaps approaching 1 million years or so, and study human ancestors. And ending then, I hope I have sort of shown you that if you're interested in human evolution, it's quite useful to have a good quality genome of our closest evolutionary relatives, because you can identify what changed here recent in human evolution, what changed further back. In the future, when we have more Neanderthal genomes, we will also be able to see on what's typical of Neanderthals. But in the long run, it will not be enough with just ancient genomes. We will have to do functional work if we really want to understand human origins. And that will then be done by iPS cells, modified iPS cells, but also, I think, with humanized mice. And with that, I then thank you for your attention. There are many, many collaborators, too many to mention. Many people have helped analyzing the genomes. Just mention one person, Matthias Meyer, who developed this single-strand library method I mentioned. Without his work, we would not have these high-coverage, high-quality genomes of Neanderthals and Denisovans today. And I thank you for your attention.